Welcome everyone to today's CFA Colloquium. So today Roger Summons is visiting us all the way from MIT. So we're very grateful to him for making this long journey and under winter conditions. So thank you. So at MIT, Roger is the Schnumberger Professor of Geobiology, and his research, the way I know Roger is through his interest in origins of life and also in characterizing Mars. So we work together on the Simon's collaboration on the origins of life. But Roger has a broad interest in geochemistry and geobiology, so in any way that life shapes a planet and leaves imprint in the planetary record. And today he will be telling us about dinosaurs. So Roger, the floor is yours.
Thank you very much, Karen, for the invitation to visit this institution. And thank you for coming to my talk. So this is the topic of the main subject I'll address today, but Karen suggested I should give you some introductory slides to provide some context about what I do and how I do it and why I do it. So this is a photograph of a place called Marble Bar in the Pilbara Craton of Western Australia. It's one of the hottest towns on Earth. You just don't want to go there in the summer. Average summer daily temperatures would hover between over 45 Celsius.
45 centigrade.
Yeah. Okay. Thank you. And it's known for this particular rock, which is called the Marble Bar Chert. It's a jasper deposit. It's essentially pure silica with iron in it that gives it the red coloration. And people argue about what's the origin of the jasper deposit, but it seems to be some kind of hydrothermal silica deposit. Not far away is a place called the Shore River, which is where we have outcrops of the world's oldest known stromatolites that people will not argue about as being the remnants of biology. There are older stromatolites that are in much worse shape and don't have the context of this one here called the Strelly Pool Formation. And this, I think, object in the photograph illustrates how you bring different disparate bits of information together to try to understand a very complex issue.
Why is the penknife in the picture?
That's for scale. I should actually have a centimetre scale there, but I had the penknife. Don't have it anymore, sadly. I got lost it in Antarctica. Anyway, so the stromatolite is, what have I done? I'm back. Okay. Sorry about that. So this is the stromatolite, and it's layers of calcium carbonate laid down by some kind of green slime, some kind of sticky microbe accumulating sediment. But it's growing on boulders of that rock I showed you before, the Marble Bar Chert. And the Marble Bar Chert boulders have rounded edges. And in order to get like that, they had to be weathered out of a deposit and tumbled to round off the edges. And then the stromatolite grew on top, and that required sea level change, and it required sea level change to first expose it and weather it, and so there's a significant amount of time embedded in this object, firstly to weather out and round the marble bar chert, but then for the stromatolite to grow. So it's evidence of growth on shallow rocky platforms where we find stromatolites today in certain water bodies. So it could be, in fact, the oldest direct evidence of a sea level change. But also, we think these things formed as a consequence of photosynthesis, and I think it's the most logical explanation for the formation of these things, is they're driven by photosynthetic microbes. So we have geological data. We also have geochemical data. So there's chemistry embedded in that stromatolite. There's organic matter. There's a small amount of pyrite. There are chemicals in here that also contain evidence of having been processed by biological activity. So we have two records of Earth history. There's one I've just shown you, which is geological and geochemical data, and it's preserved in rocks, and the rocks ordered according to the oldest below and the youngest above. But we also have the information that's encoded in the fossil record, represented here by the sort of genetic diversity of modern organisms that people have classified according to the earlier rules of classification, and we're using the occurrences of these organisms in rocks of different ages to infer something about the trajectory of biological evolution. But I think the last 25 years have brought a spectacular new tool to the table, and that is using the genetic or genomic data encoded into modern organisms to put another, I guess, level of sophistication on how you can interpret data. Because all life on Earth is descended from a single common ancestor, we believe, and all subsequent life is encoding some aspect of the history of its evolution. So we have geochemical data that you get from rocks, and we have genomic data that we can get from modern organisms, and then we have independent support for testing different biogeochemical hypotheses. So the techniques we use, of course, the geology I said before. So these are much younger stromatolites. You see they're very big domes. They're sort of car-sized objects from Central Australia. The chemistry of those rocks tells you something about the chemistry of the water from which the rocks were deposited. So we've got fluid chemistry that you can infer from those rocks. What I study, and I'll spend most of the rest of the talk discussing, are the structures of lipids, which are recalcitrant biomarkers and have a record going back 1.6 billion years as far as we know. These sorts of molecules and the inorganic compounds that we can find from rocks. The molecules, for example, contain carbon and hydrogen, which have isotopic ratios that tell you something about the processing that led to the formation of that molecule. And of course we have environmental DNA now that people can isolate from different places and sequence and put together to get a picture of, through the DNA, of what's living in that environment. And all these techniques are complementary. You should be able to make a story that makes sense integrating that sort of information. I don't know, does anyone recognise this place? But, yeah, exactly. It's the Roman bath in Bath, and it's formed by a sort of geothermal spring that has all sorts of interesting things living in it. I guess it was thought to be beneficial to people's health, maybe is. But there's an organism that was isolated from that place called Methylococcus capsulatus, and it's an organism, and it's the Bath strain, the one that the original strain came from bath. It's a lab rat, so you can get it from a culture collection and study it, and everyone can get the same organism and study it in different ways. But its physiology is to eat methane using oxygen. And as a consequence, because it's eating methane typically has a very depleted carbon isotopic signature. It's the most distinct molecule on the planet isotopically. Anything that processes that methane carries that isotopic information. So this organism makes very, very distinctive lipids, which we can find in rocks, and when we can find them in rocks, we can see exactly the same isotopic pattern that we would find in any modern system. So it's a combination of these things of using microbial biochemistry, the known physiology of the organisms, the genomics, and the molecules that they make that allows us to, gives us a piece of information that we can use to go back and query the ancient record. And we have, so I'm sort of focusing on molecules, but we have other kinds of fossils that we can also look at, like biomineralized tissue, so bones, relatively easy, bones and shells are easily preserved. Sometimes, however, fossils become permineralized, so this is pyrite that's replaced the soft tissue of a trilobite, and of course we have organic molecules. So we have all these things that we can integrate and query, try to make sense of. So coming back to the dinosaur, this is where I'm going to take you. It's kind of weird, probably, but it hopefully will make sense by the end. So we've looked at the geochemistry of this fossil dinosaur, and we can see aspects of its pigmentation that are informative, but we also got sample of the gut contents that had steroids in it. And from the distribution of those steroids, we can infer that the dinosaur had a microbiome, so it had internal flora helping it process its food. And it's processing steroids in ways that we do ourselves, as well as primates and our ancestors, Neanderthals, for example. And then I'll talk about this issue of how the modern gut microbiome, how you can use information from the fossil record that helps you interpret something about the modern gut microbiome, and potentially of use in understanding the fate of the gut microbiome in the context of industrialization, which is a serious issue. Anyway, to get back to the dinosaur, so my colleagues reported a new exceptionally preserved ankylosaur. This is an armoured vegetarian dinosaur with horny armour and well-preserved scales. It was red-coloured, and from the distribution of its colour you can infer that it was counter-shaded, that it was dark on the top, light on the bottom. Lots of animals use that as a technique to avoid predation. And this fact that it's camouflaged suggests strong visual predation. So in pictures, so here's a photo of the dinosaur showing the preservation of scales and the exceptional preservation in 3 dimensions. The chemistry of these scales tells us that one component was a molecule called pheomelanin. Pheomelanin is the coloration for red hair, as opposed to eumelanin, which is the coloration of black hair. And the fact that we can see pheomelanin tells us this dinosaur was probably orange-brown coloured. So this particular fossil has an interesting history. It was discovered in northeast Alberta when a guy driving a bulldozer was excavating the sediment above the tar sands. So they mine the tar sands by strip mining, but they've got to get rid of the surface sediment. And he disturbed this particular fossil, and it's hosted in a thing called the Cretaceous rock called the Clearwater Formation, which is sitting above this McMurray Formation, which is the one they're mining. The depositional environment of this formation is nearshore and marine environment. The specimen lacked any evidence of scavenging, and it was encased in a large iron carbonate concretion. It seems like that the animal was bloated, but then when the gut contents were released, the thing filled up with sand, which enhanced the 3D preservation. And this concretion must have started to form very quickly because there's no evidence of any predation. And there's fabulous photos if you're interested to go to the, I think it's a National Geographic website. Spent a lot of time documenting this particular fossil, but you can see this is the head showing these armoured plates and horns on the back. Another photograph showing the armoured horns. Normally these things decay. What you're left with is just the bones of a dinosaur. So the fossil, in fact, was so spectacularly well preserved that it's incredibly informative about what these organisms or what these animals originally looked like. Again, plates all preserved in situ. So the aim when we started this piece of work was we were given samples of the plates, and the idea was to do organic geochemical analysis to see if we could say something about the pigmentation. And overall to try to understand the processes leading to such spectacular soft tissue preservation. So here are some horrible chromatograms, but I have to show this is how we do our work. We take sample, an incredibly small sample, and pyrolyze it, so heat it in a stream of helium to about 700 degrees. The molecules are polymerized, so it's a macromolecule, and what we're looking at here are the fragments generated when you actually heat it under those conditions. It generates small fragments, but from those fragments you try to put together a sense of what the original composition of the scale was. So here is blow up of one of those samples. It's a horn sheath, and all these numbers here correspond to individual molecules that we can identify. And it's this molecule here, 14, the peak mark 14, that turned out to be a molecule called benzothiazole, and it's incredibly rare, and we think that's the biomarker for pheomelanin. So here is chemistry of melanins. The eumelanin, the black pigment, is a polymer of indole-like molecules, and this is pheomelanin. It's rather similar, but the difference is it contains a number of sulfur atoms. So it's the presence of this benzothiazine, which is the decomposition product of this, that tells us, that makes a direct connection between the pyrogram and a particular pigment. But that work was published several years ago, and in the interim we were given a sample of the gut contents of the dinosaur, which you can see here. And these are gastroliths, so they're pebbles or concretions inside the gut of the dinosaur, like birds have sometimes swallow pebbles to help them digest their food. So it's the remains of the dinosaur's last meal, presumably, as well as these gastroliths. And we did the same approach. Here's a pyrogram, and 14's there, but it's in the grass. So the pattern is very different to the ones for the outside horns and scales. I won't try to explain the origins of all the other peaks, other than to point out there's this little cluster of peaks at high molecular weight. And when we look at them using specific kinds of mass spectrometry, we see actually 4 peaks are present. And being chemists, we're interested in what's the identity of those peaks, which you can determine from their mass spectra. I'm going to bore you with some mass spectra next. But from the mass spectra, we can infer that these 2 peaks correspond to steroids with 27 carbon atoms, and these 2 correspond to steroids with 29 carbon atoms. So what's the significance of that? Well, the significance is that these 2 are derived from cholesterol, which, being an animal, its main steroid is cholesterol, and these are plant sterol. So this is residue of the plants that the ankylosaur was feeding on. But I've given these notations here, 5 beta and 5 alpha. That refers to the stereochemistry of these particular steroids, the three-dimensional aspects of the steroid that tell you something about how they were formed. And mass spectra, I won't go into any detail there because it's complicated, but what we can say is one of the beta one has its hydrogen at this position at 5 coming out of the plane of the screen, and this one, 5 alpha, it denotes the same molecule, everything else identical, but the hydrogen at this position goes behind the plane of the screen. So what would the significance of that be? Well, the significance is this is cholesterol. When this is hydrogen, the molecule has 27 carbon atoms. We wouldn't be out here. I like to tell people that we wouldn't be here without this molecule because it's central to how our metabolism and how our particular cell walls operate. It's also important as a signalling compound. So people have been studying steroids preserved in sedimentary rocks for probably 50 or 60 years, and we know an awful lot about it, I think. But what happens to cholesterol is this double bond here gets reduced, and if it's reduced typically in your gut, it will form this molecule here, coprostanol, and we think the fossil analogue of coprostanol is coprostane, this molecule here, which is the one we find in the ankylosaur. Alternatively, if this is reduced by pure chemical means, or in some cases by microbes, it goes to a more thermodynamically stable form, which is the 5 alpha. And this fossil is 5 alpha. So we've got in the dinosaur, we've got both the 5 beta and the 5 alpha, and the presence of the 5 beta, I think, tells us that that steroid was converted from cholesterol by a microbial consortium similar to the ones we have in our guts today. So we think this coprostane is a diagenetic product of coprostanol, which in itself is a reduction product of cholesterol. And the proportion of the 5 beta to the 5 alpha we observe here is only found today in the feces of humans, great apes, and some other large animals. It's produced by bacteria that live in the gut, and the conversion has been proven to take place by some bacteria isolated from the gut and experimented with in the laboratory, right? And because it's so specific to feces, it's widely used as a biomarker for pollution by fecal pollution. So, for example, when there are storms and sewage systems overflow, you can evaluate the extent of the pollution by using the coprostanol. Okay, it's primarily related to gut flora, and its formation appears to be controlled somewhat by pH and the time that the food is in the gut. And the exact same processes take place, the plant sterol, which is stigmasterol. And so we saw similar ratios for the animal's own steroid and the plant steroid, reflecting processes that would go on in our gut today. The significance, I think, part of the significance of coprostanol is because once your gut flora reduces the cholesterol in your gut to coprostanol, it can no longer be reabsorbed. So if you have a microbiome that's very effective in converting cholesterol to coprostanol, that correlates with having lower blood cholesterol levels. And the rest of the talk is pertaining to this particular issue. So we know that cholesterol is removed by conversion to coprostanol. It's been shown for many years, for example, in this paper about lactobacilli. It's been demonstrated in the lab by this organism, bifidobacterium, isolated from human gut, can convert cholesterol to coprostanol. So there's plenty of experimental evidence that this is a real process. And why it interested us in, as I said, people have been studying the presence of steroids in ancient rocks for many, many years, and we think we know all the processes by which steroids get modified and preserved in rocks. And I know of only one other instance where you find sediments with a high proportion of these beta steroids related to coprostanol. So the very the discovery of that stereochemistry I thought was particularly significant. As I said, also fecal sterols are widely used as a way to monitor pollution. So this is an old paper from former colleagues in Australia studying the composition of the steroids in feces of different kinds of animals. You wonder why people would do this, but these are steroid distributions in cows, horses, sheep, possums, cats and dogs. And for example, dogs do not make coprostanol. So when you see fecal pollution, you can exclude the possibility that it was the dogs that people were walking on the sidewalk. That fecal pollution potentially is mostly human, the pollution that we're particularly worried about. Although some kinds of animals, cows will produce a small amount of these 5-beta steroids. But you can use the distributions of the steroid carbon numbers and the steroid stereochemistry, and you can apply statistical techniques, and you can map out distributions that tell you for sure whether particular sorts of feces came from particular kinds of animals. So cows and sheep plot in a particular area, horses, hens, pigs, for example, all distinct from humans, which are plotting down in this bottom of this quadrant. And the purpose of showing that is that with diagrams like this, you can identify feces as human and as distinct from feces of animals. And why is that significant? This is Ainara Sistiaga, who was formerly a student in my lab, visiting student from Tenerife in Spain, and then she came back on a Marie Curie fellowship and spent several years as a postdoc. And she's she studies prehistory. She's particularly interested in Neanderthals, and here she is as a student, and she's excavating what was a fireplace in a Neanderthal habitation site on the Iberian Peninsula. So she's excavating the fireplace, which presumably was used for cooking, but it seems like it was also used for pooing. So El Salt is part of many sites on the Iberian Peninsula with a lot of sediment accumulated, and the sediment was laid down during its habitation by Neanderthals. And one of the objects that she recovered from there was a coprolite, and evidence that it's a human coprolite, the first piece of evidence was from microscopy, where they could identify objects that were very reminiscent of gut parasites. So parasites known to be associated with the human gut was the first clue that this was a human coprolite. But when we look at the steroids present in that you could extract from this object, we have coprostanol, cholesterol. The ratio of those two peaks are a bit similar to the ones I showed you from the dinosaur. These are the sterols, not the steranes, as well as the steroids with 29 carbon atoms, which are the ones that are coming from the plant steroids. So first piece of information you can draw from this is that Neanderthals ate their veggies. And this is probably this most... I wouldn't know the percentage, but some of this would have been made by the person whose poo this belonged to. But some of it would also have been from the diet. So in order to exclude the possibility that this coprolite actually came from animals, and the most likely source of a coprolite like this would be one of the great apes, Ainara also did a study of poo from chimps, gorillas, horses, chimps, and applying the same kind of principal components analysis came up with this plot based on the distribution of steroids. These are all the samples of great apes, and clustered together are the Neanderthal coprolites and human coprolites. So based on the presence of a parasite for humans as well as the steroid distributions, it's pretty convincing that the coprolites were in fact of human origin. Somewhat later, Ainara went to this site in Portugal, another well-known site of Neanderthal habitation. That's a picture that is taken on the way to a cave, Lapa de Piqueiro. And here's the excavation site, and people excavating at different levels. And because of the work she'd been doing on El Salt, she was invited to participate in this excavation. She went on a field trip there. When it was on the first day that she was there, she's recognised, I know what that is. And these are three distinct coprolites, evidence perhaps of a latrine, because when you look at the steroid patterns of those three coprolites, they're all different. Now, two of them, this one here and this one, 2 and 3, look similar. I mean, there are significant differences, but they look similar enough that you couldn't distinguish them. But this one is so different, with so much more coprostanol compared to cholesterol. This must be from a different individual. And finding those coprolites together suggests that there was part of a latrine, or that came from a latrine. So, from the ratios of the different steroids, the carbon numbers, you can infer that this is human fecal matter, and the different ratios of conversion tell you that you can only get this particular occurrence when you're dealing with different microbiomes. So this leads us to what Ainara did next in collaboration with members of Eric Arm's lab in biological engineering at MIT. He runs a program of fecal transplants, that is, getting donations of feces from healthy individuals, transplanting to people who have health problems that might be solved by fecal transplant. And his two postdocs, Mathieu Groussin and Mathilde Poyer, and Ainara came up with this idea of starting the global microbiome conservancy project. They started with no funding, but the idea came about from recognising that people from with non-industrialised lifestyles have different bacterial diversities, and they also have different abilities to convert cholesterol to coprostanol. But the fact that so many populations of people living in remote areas are under stress from people from industrialised lifestyles, or the imposition of industrialised lifestyles on people with more natural lifestyles, meant that the diversity of the biochemical and the organismic diversity of the gut microbiome is also under threat. So they set out to achieve incredibly complex task of getting people from, like this person is here is a Hadza from Tanzania, and these people, their traditional lifestyle is to eat carrion, so they feed on the corpses of dead animals, have a very unusual gut microbiome, as you might expect. And the idea was to convince these people to give both blood samples and samples of faeces that could be preserved. The samples of faeces can be sequenced to get information about the diversity of the microbiomes, but the fact that they have archive samples of this might be useful in the future for understanding evolution of gut microbiome associated with industrialization. So Ainara and her colleagues managed to convince people from a number of remote communities to become engaged in this process. I don't know how you go about asking somebody that you have difficulty communicating with to give you a sample of their poo, but they did it. And here's some data that is in a paper that's just been, submitted for publication. It's just again, principal components analysis showing the bacterial diversity of people from, so each dot is an individual, and these are people from different communities, and note that the black dots here correspond to people from the local community. I think I'm one of those dots. And so these represent urban communities with very different bacterial diversities in their guts to people from rural communities, for example, those in Africa. All these samples have been analysed for their steroid contents, conducted in the way that we would have done with a Neanderthal coprolite. You have the fecal sample, you extract it with organic solvents, you use those solvents to separate the non-polar molecules like the steroids, you derivatize them, you analyse them by gas chromatography, mass spectrometry. Similar way, many ways similar to the way we looked at the dinosaur skin, and you get a picture of steroid distribution, and this one clearly is an individual with a very high content of plants in the diet. It's completely dominated by the C29 steroids as opposed to the C27 steroids. And with that sort of data, you can, and this is a log plot of the conversion of coprostanol to cholesterol. So these are non-human primates. So this is the data from gorillas and chimps. This is the data from the 3 or 4 Neanderthal coprolites we had access to. And this is the span of data, much greater, of course, for non-industrial samples from people from non-industrialized populations. And this is the data from US donors. And you can see a much wider range in conversion, but many people or donors with very, very poor conversion of cholesterol to coprostanol. So here's a comparison now of the proportion of bad converters in US donors, for example, compared to people with non-industrialized lifestyles. Here's some more data, similar kinds of data. This is the ratio of 27 to 29. So clearly people from urban lifestyles are having much higher levels of C27 steroids, that is cholesterol, in their diet. Again, coprostanol to cholesterol. People with urban lifestyles tend to have a lower conversion rate. And this is the overall ratio of 27 to 29. That is animal steroid compared to plant steroid. So people with non-industrialised lifestyles tend to probably have more diets that are more beneficial to conversion to of cholesterol to coprostanoid and potentially having lower levels of blood cholesterol, which potentially is seen as a health benefit. Anyway, this is the paper that my colleagues have been trying to get, or presently trying to get published on that topic. And I'll just finish by acknowledging people who did most of the work I talked about. Ainara did the work on the Neanderthals, chimps and humans, and Joshua Herrera, he was a master's student in LA, did the work on the dinosaur fossils. So thank you for your attention.
So questions for Roger?
How do you date the coprolite? Do you do it from the context, or is there a way you can date it directly?
Context, it's the carbon there is beyond the point at which you could use carbon-14. I'm not an expert on how they date those things, but 80,000 I think is the site in Portugal. I think the number might have been up there, but I honestly don't know how they do that, but people are confident, I think, of those ages.
So, okay.
So a question from up here. So you showed the distribution of gut biomes for humans.
Yeah.
Do you know how much individual humans change on that diagram depending on what we've been eating, say, birthday cake versus a spinach salad, or?
So it's in the paper, and from the top of my head, what I can recall is that people from with the industrialised lifestyle, that is, say donors from Boston, have greater biodiversity in the gut microbiome, and greater evidence from that biodiversity of horizontal gene transfer in the gut microbiome. Whereas the Hadzes, for example, had overall lower biodiversity, and evidence of lower amounts of horizontal gene transfer. So I don't know what the significance of that is because I'm not a biologist, but it seems to be that it's significant that people who tend to be good converters of cholesterol and coprostanoid would have a less diverse gut microbiome.
Do you notice any difference between the microbiomes of Neanderthals and humans that are independent of their their location, their diets? In other words, any evolution?
Well, the problem is that we can only look at molecules that are relatively recalcitrant, and the steroids are relatively very stable. So once everything else is gone and you're in a puddle of stuff in a coffin, presumably it's fat, and most of it's going to be those fat molecules that are the least able to be destroyed by bacteria. But DNA, which you need to evaluate the bacterial diversity, that's incredibly fragile, and that would go within hours of the coprolite forming. So you can only really do DNA analysis on fresh samples of feces that have been properly preserved. So we can't evaluate anything about the microbiome of the Neanderthal other than looking at these steroids that tell us they did have a microbiome. But and you can look at the steroid contents as some evidence of the diet, but we've got four or five coprolites. It's not really enough to do a super detailed study.
I'm just wondering whether you could make any distinctions in the evolution of Neanderthals from humans.
I think that'd be a big ask.
Sure.
Actually, a naive question. When you do the mass spectroscopy, you seem to be very confident in identifying the molecule. Are you just on the molecular weight?
No.
What else do you have?
You have a fragmentation pattern. So there's certain kinds of mass spectrometry that just pick up molecular ions. So MALDI, people use MALDI-TOF to map particular compounds by their molecular ions on a surface or in a material. But what we're doing is gas chromatography and mass spectrometry, which involves separation of the compounds. So we have information on retention time, that is the time from, so this is a time scale on the bottom, so this is the time after which you injected the sample on the instrument to when it came off. So that tells you something about molecular weight and about polarity. And under each of these peaks, we have a series of mass spectra that have a fragmentation pattern, which is essentially a fingerprint. And I did show it when I showed the early pictures of mass spectra, but I should have explained that the spectrum itself is a fingerprint, and different steroids will have subtle differences in the mass spectra that allow you to say something about stereochemistry. We're confident, we also use authentic standards when we're analysing these things. So, for example, petroleum is a wonderful thing. Besides powering our cars, it's a complete zoo of interesting molecules. And once you've gone through and identified a lot of things in petroleum, you can use a petroleum as an authentic standard for fossil hydrocarbons.
Yeah, sorry. You had the interesting thing about the industrial versus the non-industrial. That was really good. But you had that cloud of distribution, and it looked like there might be some subgroups. I wonder if they correlated with different types of diets within those.
Yeah, I'm sure. And that, I think, is in those kinds of plots are usually three-dimensional, and you can rotate them, and from those rotations and from the scores and loadings that backs up that diagram, you can identify the groupings, and you can identify why a particular group is clustering together. But that particular cloud doesn't show that detail. But the detail is in the diagram.
So, but is a sort of follow-up going to look at what the subgroupings might mean?
Mediterranean diet?
That's what I was thinking, yes.
Maybe
Well,
but
the guys who eat carrion might have a very different biome from the people who are mostly gathering fruits and nuts.
Yeah, absolutely. But I couldn't tell you what the clustering meant. Our contribution was generating this sort of data, and it was the engineers that did the DNA sequencing. You can see from the number of people on the paper how much. Yes. There's an enormous number of different skills involved in generating a data set like this. And I can only claim to be near the end on the basis of having provided the facilities to do the analysis.
Are there other gastrolithic coprolite samples from other dinosaurs, other types of dinosaurs that have been examined?
Not as far as I'm aware, and
so you look at what theropod stomachs were like, or sauropods.
if you could find specimens that had been as well preserved as this, but I think that's going to be an incredibly rare occurrence. The fact that typically the ankylosaurs have lost all their scales and horns, all their external covering, when presumably relatively unrelated microbes are exchanging DNA, usually through some other vector like a viral particle infecting one kind of bacterium, and then those viruses being picking up some DNA from that organism, being released into the system again and being picked up by another organism, a different organism, and sharing the DNA that it got from the previous one. So it's a process that's going on all the time. It's presumably a process that has shaped us as human beings. And in the lunchtime talk, I showed evidence of horizontal gene transfer of the genes responsible for making these steroids. And you can use those horizontal gene transfers, map them back in time, and you can use molecular clocks to put some timescales on that, when those particular transfers took place. So organisms on this planet have been sharing their genes forever and ever, as far as we know. And I think one of the main